Welcome to the deep dive. Today, we're looking at something that seems, well, incredibly specific. We've got the personal gardening notes from someone who was documenting their experiments growing a single plant.
Right, the Peacock Flower, Caesalpinia pulcherrima, and, that specificity is actually what makes this so great.
How so?
Well, this isn't some dry textbook. It's a real field journal. You can see the trial and error, the genuine curiosity, and some really surprising discoveries.
And we're going to pull universal rules out of this, things that apply to basically anything you grow, whether it's flowers, vegetables, even trees.
That's the idea.
So that's our mission for this deep dive. We are sifting through these detailed observations to find really actionable knowledge for you. Specifically, we're unpacking two big challenges the author ran into right at the start.
The first is that age-old mystery of delayed germination, dormancy.
And the second is figuring out the absolute perfect time for transplanting to make sure you get the best possible growth. Getting these two things right can really take your gardening to the next level.
It really can. So let's start where the author did, right at the very beginning, with the seeds. The first notes, well, they sound a little frustrated.
Oh, absolutely. The observation was really simple. A batch of 10 seeds, all planted at the same time, identical conditions.
Okay.
And 4 of them germinated almost immediately. A few days later, boom, there they were. But the other 6?
Nothing.
Nothing. Just sitting there. Then 2 weeks go by, there's this huge long downpour of rain, and suddenly 2 more seeds pop up. Just like that, overnight. It totally caught the author by surprise.
And that is a classic botanical mystery. I mean, you have 6 seeds, all in the exact same soil, same temperature, same little microclimate.
Right.
And yet some sprout right away and others just wait. They wait for this, what seems like a totally random event, like a ton of rain. So it raises the question that the author was trying to answer. What is telling these specific seeds to wait while their siblings are already growing?
And the research that the author did pointed to one culprit, at least for the peacock flower. It's a mechanism called physical dormancy.
Which is often categorized as exogenous dormancy.
Exogenous.
It just means the problem is coming from the outside of the embryo. And that word physical is really the key here. The problem is mechanical, not biological, at least not at first.
Okay, so what's the mechanical problem?
It's all about the seed coat, the shell. For the peacock flower, and actually a lot of other species, that seed coat is designed to be incredibly tough. It's impermeable.
So it's like armor, not some kind of internal clock telling the seed when to wake up.
Exactly. Think of it as a tiny, perfectly engineered defensive capsule. This hard shell physically blocks the two things that are absolutely critical for starting life.
Water and oxygen.
Water and oxygen. If water can't get through that coat, then all the metabolic processes inside the seed that need water, the swelling, the enzymes firing up, they just can't start. The plant is stuck in stasis.
Okay, that completely explains the sudden sprouting after that big rainstorm. It wasn't the seeds deciding, now's a good time. It was the constant soaking rain that finally just wore down or weakened that tough barrier just enough for water to get inside and kick things off.
It was a physical breakthrough, exactly. Not some kind of conscious decision from the plant.
And when you think about it evolutionarily, that makes a ton of sense, right? Why would a plant develop this?
It's a survival strategy. It's insurance. If you're in a dry or unpredictable climate, you can't risk germinating after one little passing shower.
Because you might sprout and then the soil dries out a week later and you're done for.
Precisely. The hard coat ensures the seed waits for sustained moisture, the kind of long, deep rain that signals the conditions are right for long-term survival.
Which means the gardener can step in and sort of play the role of that sustained rain. If we know the hard coat is the barrier, the solution is pretty obvious.
And very effective. The author's notes are really clear on this. For seeds, have a hard coat, you can just bypass that whole evolutionary waiting game by giving them an overnight soak before you plant them.
Just in plain water.
Yep, just soak them overnight. This process, which is a form of scarification, it artificially softens that coating. And it can take your germination rate from, say, 40% up to 90%, or even higher.
Now, here's a question. If we're interfering with a natural protective process, is there any downside? Are we risking anything by soaking them?
It's a really good question. The main risk is actually over-soaking. If you leave them in water for multiple days without changing it, they can rot or even drown. Well, once their metabolism kicks in, they need to respire, and they can't do that properly underwater for too long. But a simple overnight soak, 8 to 12 hours,
Yeah.
That's usually the sweet spot. It's enough to soften the shell without putting any real stress on the embryo inside. It's just a smart shortcut.
Okay, now this is where the notes get really useful for everyone. The author realized this wasn't just a peacock flower issue. They started noticing the same kind of delayed sprouting in other common garden plants.
Yes, the list in the notes mentions vinca, which is periwinkle, basil, and, interestingly, portulaca, moss rose.
Wait, basil? That's surprising. We grow basil all the time, and it seems to pop up pretty quickly. Why would a leafy herb like basil be on a list with these other tough ornamental flowers?
That's a great point. Basil doesn't have the same kind of rock-hard physical dormancy as a peacock flower, but its seeds still benefit a lot from a soak, especially if you're using seeds that are a little older.
So even if the barrier isn't as extreme, the principle is the same.
Exactly. If you've ever had patchy basil germination, where some come up and others don't, a pre-soak is often the ticket to getting a nice, full, uniform stand. It just proves the universal idea. If germination is slow or erratic, think about the coat.
So the big takeaway from this first part is that instead of just waiting for nature, we can proactively help the seed get the water it needs. We just have to overcome that physical barrier.
That's it. So now let's pivot, because getting the seed to sprout is only half the battle. The notes show that what you do next and when you do it is just as critical.
Right. This brings us to the author's really simple, but I think very revealing experiment about transplant timing. They had 4 healthy little seedlings, and they split them into 2 groups.
2 of them were moved right away into bigger, roomier pots with fresh soil. The other 2 were just left behind in their original tiny seedling tray cells. They became the control group.
The let's wait and see group.
Exactly.
And the results, I mean, the photos and the notes are just shocking. The 2 seedlings that were transplanted just took off. They exploded. The notes say they grew significantly taller and larger. They look so much healthier.
And the 2 that were left behind?
They just stalled. They look small, kind of sickly, completely stunted. I mean, they were failing, even though they got the same sun and water as the others.
And the reason for that huge difference is, well, it's something every gardener needs to understand. The seedlings left in those little cells had become root-bound.
They just ran out of space.
They ran out of space.
Yeah.
And when the roots can't grow outwards, they hit the plastic walls of the cell and have only one direction to go.
Around and around.
They start circling the bottom. And that circular pattern, it's a disaster. It's like a slow strangulation. The roots can't spread out to find new nutrients, and more importantly, they can't expand their surface area to absorb water efficiently.
So the whole root ball just becomes this dense, tangled mess. And even if you're watering it and fertilizing, the plant just can't take it in. It's essentially starving in place.
And here's the really bad part. Once a seedling gets severely root-bound, that damage can be permanent. You can try to tease the roots apart and transplant it later, but that plant may never, ever reach its full genetic potential.
You lose time, and you probably lose yield or flowers down the road.
For sure. That's why the timing is so precise. You can't just go by a calendar or say, oh, I'll do it in 3 weeks. You need actual signs from the plant itself.
And that's what's so brilliant about the notes. The author developed this really simple 2-part system to know exactly when a seedling is ready. It's called the leaf rule and the root rule.
And you really need both. One tells you if the plant is mature enough, and the other tells you if it's structurally strong enough. Functional maturity doesn't guarantee structural stability, and vice versa. You need both before you put the plant through the stress of a move.
Okay, so let's break down rule number 1, the leaf rule. This is a sign of functional maturity.
Right. And for this, you have to know the difference between the 2 types of leaves a seedling makes. The very first ones to appear are the cotyledons, or seed leaves.
They're usually kind of small and round, right? Not very distinctive.
Exactly. Their only job is to get energy from the stored reserves in the seed. They're like the plant's training wheels. But they are not the sign you're looking for. The key indicator is the appearance of 1 or 2 pairs of true leaves.
And true leaves actually look like tiny versions of the adult plant's leaves.
That's it. You see them. That is the big metabolic milestone. It signals that the plant has graduated. It can now perform photosynthesis efficiently.
It's not living off its seed lunchbox anymore. It's making its own food.
It's making its own food from sunlight. It is functionally independent. And once it hits that point, it's mature enough to handle the stress of being transplanted.
Okay, so functional readiness, check. We've got true leaves, but we still need to make sure it won't fall apart during the move, which is where transplant shock comes from.
Right. And that's where we get rule number 2, the root rule. This is all about structural integrity, and it does require a quick, Gentle, physical check.
What's the prep for that?
The preparation is actually super important. You want to water the plant about an hour before you do the check. That moisture helps the soil and all the tiny new rootlets kind of cling together. It makes the whole thing cohesive, like a little damp sponge.
And then the technique itself is really specific in the notes. There's a big don't.
A huge don't. You gently try to lift the entire seedling out of its cell by a leaf, never ever by the stem.
Why not the stem? It seems like the strongest part.
It seems like it, but it's actually the most fragile. The stem, it's the tiny pipeline between the leaves and the roots. If you pinch or crush it, you can kill the seedling instantly. A leaf is much safer. You get leverage without damaging that critical connection.
Okay, so you lift it gently by a leaf. What are you looking for? What's the sign that it's ready?
It's very simple. If the root ball stays intact, if that whole little cube of soil comes out in one solid piece, then it's structurally ready to go.
Got it.
It means the roots have grown enough to create a solid network that holds everything together. That integrity is what minimizes transplant shock, because the roots stay protected in their soil and can get back to work immediately in their new pot.
That makes perfect sense. You're trying to keep that whole little ecosystem together, but the root roll is also a warning system, isn't it?
It is. It's the final alarm bell. If you lift the seedling and see a bunch of roots circling the bottom of the cell.
The ones we talked about earlier?
Right. If you see that, the plant is already on its way to being root-bound. You need to transplant it immediately, no delay. Give it a chance to straighten out those roots before the damage becomes permanent.
So it's this dual check. You need both functional maturity, the true leaves showing it can feed itself, and structural integrity, the intact root ball showing it can survive the move.
You apply those two rules, and you are protecting your seedlings from the two things that kill them most often: starving because they were moved too young, or strangling because they were left too long.
And I think we've all had that experience. You buy a six-pack of tomatoes from a big store, they look okay, but you plant them and they just sit there for weeks.
Yes. That's often because they were already severely root-bound in that little plastic pack. You're starting the race with a huge disadvantage because they missed their perfect transplant window a long time ago.
So we started this deep dive with some really specific notes on one flower, the peacock flower. But by digging into the why behind the author's experiments, we've pulled out these two universal sets of rules.
Yeah, how to deal with physical dormancy by soaking the seeds, and then using the leaf and root rules to find that single perfect moment to transplant.
It's a great example of how just being curious and paying close attention can lead to mastery. These rules really cut through all the noise and give you clear, actionable steps.
They do. They help you beat the seed's physical shell, and then they help you beat the seedling's tiny plastic container.
It's just fascinating how a few seeds and a simple side-by-side comparison can reveal these global principles that can make every single plant we grow more successful.
Absolutely. And, if we think about that deeper science for a second, we've established that exogenous dormancy, the kind we talked about, is caused by a physical barrier, that hard seed coat.
Yeah.
It raises a really interesting follow-up question for you to think about. For all the seeds that don't have a hard coat, their dormancy is endogenous, meaning it's internal. So if there's no physical wall to break down, what complex metabolic process is going on inside that seed telling it to wait? And how does that internal clock decide that the environment, the temperature, the light is finally right to start growing? The mechanical problem is gone, but the biological one, well, that really is a really beautiful puzzle.